Hello, I'm Jesse, a senior research scientist and application specialist here at Akadeum. Today, I'll be showing a complete cell therapy workflow, from an instrument-free cell isolation, cell activation, gene editing with electroporation, and finishing with cell expansion. This workflow combines the Akadeum Human T-Cell Leukopak Isolation Kit GMP with an instrument-free cell separation, followed by electroporation with the MaxCyte ExPERT GTx GMP Electroporator. This integration was specifically designed to streamline your cell therapy workflows, ensuring process flexibility while maintaining regulatory compliance and leading to better therapeutic cells. So, what makes this workflow better than your status quo? It all comes down to Akadeum's unique buoyancy-based microbubble technology. Our microbubbles gently bind to unwanted cells, floating them to the top of the processing bag. This allows your untouched T cells to gently settle in the subnatant. These high viability and robust cells are then ready for your gene editing workflows with increased transfection efficiency and viability. The beauty of this approach is its simplicity. Fewer steps, a cell handling time under 60 minutes, and a scalable cell isolation that integrates directly into your existing closed system GMP-compliant workflows. Inside the Akadeum Human T-Cell Leukopak Isolation Kit GMP, you'll find our strep abdomen microbubbles, our negative selection antibody cocktail, and a buffer. For this particular workflow, you'll also need standard cell processing bags, an end-over-end rotator, and a tube welder. And of course, the MaxCyte ExPERT GTx GMP Electroporator, none of which are included in the kit. That's how simple it is to integrate the Akadeum Human T-Cell Leukopak Isolation Kit GMP with your MaxCyte ExPERT GTx GMP electroporation system. Our innovative microbubble technology enables a GMP-compliant, scalable, and high-performing 60-minute workflow that provides efficient, high-quality T-cell isolation and maintains the health of your starting sample to provide increased transfection efficiency, higher cell viability, and more therapeutic cells. Cells are now ready for your potency assays and fill and finish. To learn more or request a demo, visit us at akadeum.com.